Hi everyone on the other side of the screen. My name is Diego Grinman and I'm a postdoctoral associate at Yale University, where I study the effects of breast cancer and mineral and energy homeostasis. I'm also a member of the editorial board of HubLE, and I'm here on behalf of HubLE to interview Max, who presented his work at the ACTS 2021. Max, do you want to introduce yourself to the HubLE audience?
Yes, of course. Hi, Diego, thanks for the opportunity to have this little interview. I'm a PhD candidate in the Netherlands. I work at the Erasmus Medical Center in the lab of bone and calcium metabolism. And my project is mainly about finding gene editing approaches for a condition called craniosynostosis.
Nice. So Max, what are you presenting at the ACTS 2021?
At the ECTS, I gave a global overview of the presentation of my project, and I zoomed in on the focus on getting these certain mutations inside the genome of the cells.
Sweet, that's very cool. And what, if you can, in a sentence, what are the key findings of your work?
So we use prime editing for this project, and it's a fairly new developed technology, and we encountered a couple of issues. And one of the most interesting that we encounter is that sometimes you have this prime editing guide RNA, and it needs to be adjusted a little bit. And we found out it's not really a gray zone, but really black and white. Sometimes they just don't work at all, and sometimes if you just adjust some nucleotides, they work fine. And with a correct construct, we were able to get the mutation that we wanted by specifically adjusting the genome. We got the mutation of our interest into the cells.
Wow. Yeah, I mean, I bet you try many mutations, right?
Yes, we do. We also have some other targets, and we are very excited to also get those in our cells.
Okay. Well, and if you get to say what are the big pictures, the implications of your findings in the near term and the long term.
Yeah, so it's not only the efficiency that you can adjust with your prime editing construct. It appears that it's really important to adjust them to make them even functional. But aside from that, we are now able to introduce specific alterations of the DNA into our cells of interest, and we are limited. We are not so limited by other factors that that you can encounter with other types of CRISPR-Cas9 gene editing.
Wow, yeah, that has many implications for sure. And yeah, I would like to ask you what is next in your career? What are you going to do next? What's your next step?
So now that we already got a couple of milestones, we have a lot more perspectives and approaches. And one of those is we also have a couple of patient-induced pluripotent stem cells that we could generate. And now we also have those specific mutations, but in a, pluripotent stem cell. So we want to differentiate those as well to a osteogenic lineage and see how they differ from normal wild-type cells and how they compare to our own cells that we mutated. Okay. And if everything goes all right, maybe we can even correct the mutation of the patients and make them have a normal genome.
Gotcha. And what about yourself? I mean, personally?
For me personally, well, this is still on. This is still within the grasp of my project. So hopefully I will be able to start with this, and we're actually starting with the experiments of the iPSCs very soon.
Great to hear. So Max, thank you so much for sharing your work. And remember that if you're an early stage investigator and you want to share your ideas, we got your back. Contact us. And we in HubLE, we're going to help you with your project. So thank you so much, Max, again.
Good. Thank you for having me.
See you around.